How do cells with DNA as their instructions know what to do in order to make a specific organism? They go through the process of transcription and translation. First, we are in the nucleus of a cell, where the DNA is stored. We need to start the process of transcription. The first step is to make messenger RNA from DNA. We start off with a strand of DNA. We then write down the corresponding parts and how C goes with G and A goes with T. Then the DNA goes through the RNA polymerase, which copies a strand of DNA and makes it into mRNA. RNA does not have T's in their codes, but instead uses U's. After that, the strand of mRNA leaves the nucleus and starts heading towards the ribosome.
Next comes the process of translation, happening all in the ribosome. Before the mRNA goes into the ribosome, it cuts out unneeded pieces called introns. Introns are then removed and go into the ribosome to make the perfect piece, which are read by N3s. This is the final mRNA piece, which is all happening inside the ribosome. Each group of 3 are called codons, and the tRNA are called anticodons. G goes with C, A goes with U, and vice versa. The tRNA produces amino acids which have a code on them found at the first mRNA pieces, such as GAA. So we look at the coding and we find G, A, and A, which match up to GLU, which is on the amino acid. The next one, which is GUC, corresponds with CAG. Since the mRNA is being read first, we see that GUC is corresponding to VAL. Again, to find this, we just look at the GUC, go to G, U, and C, which corresponds to VAL. We do this with the remaining tRNA to find their amino acids. After this, the tRNA pushes one each other off to start building the amino acid chain. This is so that the tRNA is done copying and so the amino acids can start to build. After this is done, the amino acids form a chain which are all connected by amino acid strands. After translation, all the amino acids are then moved to the Golgi body complex. Inside the Golgi body complex is where all the protein is made.